When we watch a flock of starlings turn as a single liquid mass in the sky, coordinating thousands of bodies in real time, we are looking at the final result of millions of years of biological trial and error. We usually understand this incredible diversity of life through a macroscopic lens. A species faces environmental pressure, and over countless generations, it slowly adapts to survive. The engine driving that slow adaptation is the genetic mutation. Every so often, a random typo occurs in an organism's DNA blueprint, creating a tiny structural variation in the offspring. The entire standard model of biology relies on this premise. The macroscopic survival of every species on Earth depends entirely on these microscopic mistakes. But there is a mechanical problem with this model. The DNA double helix is a highly stable, tightly bound structure locked together by strict chemical rules. Because this architecture is so rigid, spontaneous structural errors should not naturally happen. The molecules simply don't have the internal energy to break their own bonds and rearrange themselves. Classical physics and chemistry identify what a mutation looks like after the fact, but they lack the mechanical explanation for the precise spark that forces the first molecular shift. If the classical rules dictate that DNA should remain perfectly stable, there must be a hidden mechanic, a ghost in the double helix, that triggers these spontaneous changes. To find that mechanism, we leave the human body behind and descend the ladder of scale, plunging deep inside a single, rapidly dividing cell. Pushing through the nucleus, we scale all the way down to the individual molecular rungs that make up the DNA ladder. Here, we find the exact physical barrier preventing mutation, the hydrogen bonds holding the two parallel strands of our genetic code together. For a spontaneous mutation to trigger, a single subatomic proton must cross from one side of this gap to the other. Yet, standard chemistry dictates that the proton lacks the energy required to jump that gap. The proton is trapped behind a solid wall. Under the classical laws of science, the mutation is physically impossible. To breach that wall, we have to look toward the erratic rules of quantum mechanics. This diagram shows a physical barrier at the quantum level. In this realm, a subatomic proton operates via quantum tunneling. Instead of acting as a solid particle with a fixed location, the proton exists as a fluctuating probability wave. Because its location is smeared out as a wave, a tiny fraction of that probability extends past the barrier. There is a measurable, non-zero chance that the proton's location exists on the far side of the physical wall. When the wave collapses, the proton doesn't climb over the barrier. It passes instantaneously through it, materializing in the new location. If this quantum teleportation occurs at the exact millisecond the DNA strands split to replicate, the chemical code is altered. The typo is locked into the sequence. A physical impossibility in classical biology is solved by the inherent uncertainty of subatomic physics. Now watch what happens when we reverse the scale. That newly altered genetic letter gets duplicated as the cell completes its replication cycle. That single mutated cell multiplies millions of times, building tissue, bone, or muscle, eventually expressing itself as an observable biological trait in the fully grown organism. The organism carries this new trait out into the macroscopic environment. If that trait aids survival, it is filtered through the process of natural selection. Over thousands of years, if that individual survives and passes the code to its offspring, this subatomic event can spread through a population. What began as a quantum shift eventually manifests as a physical advantage for an entire species. When we look back at the grand scale of human evolution, the picture looks different. We often associate the strange behaviors of the quantum realm with distant stars or high-energy labs. In reality, these same rules govern the chemistry happening inside your own cells every second. These unpredictable quantum leaps provide the exact mechanical trigger that creates genetic variety. Without them, DNA would remain locked in perfect, unchanging stasis. The long-term survival, adaptation, and diversification of all life on Earth is anchored to microscopic uncertainty.